Started. So today and Thursday, we're going to be doing meiosis. So we've talked about mitosis. We've talked about chromosomes, homologous chromosomes. We've talked about diploid cells, different cell types, all these kinds of interesting things related to cell division and cells and genetic material and DNA. So today we're going to be going into meiosis primarily, which is the division of the sex cells, gametes, producing sperm and egg cells. And we're going to be talking about the detailed process of how they are generated. And sort of, we're also going to be completing that table. If I remember correctly, we were doing that compare and contrast table I like to do between mitosis and meiosis, and finishing sort of the details of meiosis, the broad overview of the who, what, where, when, why, how. And yeah, we're going to be talking about the why. Why do we take time in introductory biology to go over meiosis, this completely different and specialized, unique type of cell division? Why is meiosis so important? And if I remember correctly, we kind of touched upon at the very end of last lecture that we're creating these genetically unique gamete cells, as opposed to mitosis, where we are at the end of mitosis, we end up with two genetically identical daughter cells to that original parent cell and to one another. And why having genetically unique cells is important. So we're going to talk a little bit about that, but mostly we're going to be talking about this intricate mechanism of meiosis and really how this unique genetic combination is created through this meiosis process. Okay, so let's begin by continuing this table that we've been doing. Should have my notes up. All right. So we left off discussing the meiosis side of things, right? And we were doing, well, let's write that first. We're discussing meiosis today as well as next class. And who, what, where, when, why, how, right? So who, what, where, when, why, and how. I believe we finished these two last time, but we'll talk about it again today, sort of review and as a preface for what we're going to be doing today. So who would be doing meiosis? What life on this planet, what organisms do meiosis? Sexually reproducing organisms. Sexually reproducing organisms. And this includes us, animals, plus all other animals, plants, and some fungi. And as we'll see, sort of biological definition of sexually reproducing, it's really just about this combining of genes and combining of genetic material from two different origins, two different organisms, combining their genetic material. That's really what we mean by sexually reproducing. And ultimately, that combination of genetic material is going to give rise and produce a new organism, like you and me. Your parents' sexual reproduction led to you and me. Combining the DNA, the genetics of your parents and mine. So these organisms do this process. And by this process, what is it? We are going from one diploid cell specifically a germline cell, and as we'll be discussing today and next time, meiosis involves two parts, two phases, meiosis 1 and meiosis 2. So meiosis 1 and then another phase, meiosis 2. So there's actually two cell divisions in all of meiosis. So that's actually going to result in 4 haploid genetically unique gamete cells. All right. Where is this occurring? Where are we doing meiosis? The reproductive organs. And for males, this is the testes. And for females, the ovaries. This process of germline cells dividing by meiosis to produce gametes happens in testes for males, ovaries for females. Interestingly enough, this isn't always occurring. It occurs at specific times in an individual's life. So we'll do when. And for males, the process of meiosis begins at puberty, adolescence. So, from there, so continuously from puberty into adulthood. So once it begins, males are actually constantly doing meiosis. I believe until we die, actually. So from puberty into adulthood, continuously generating gamete cells. Females, on the other hand, interestingly enough, females actually perform meiosis prior to birth, still in the embryo. So females prior to birth. So interestingly enough, all your for females, all the egg cells you are born with, and you're done. Technically, at puberty, what happens is meiosis 1 occurs for females prior to birth, and then it arrests, it halts at the end of meiosis 1, and then meiosis 2 occurs at puberty. But really, meiosis 1 is that critical stage where we're creating these genetically unique cells, as we'll see shortly, and that happens before you're born. It's pretty crazy. Okay. The why. So we went into some depth about why we do mitosis, right? Growth and development of an organism, repair and regeneration, asexual reproduction. The why for meiosis, I would argue, is as important, if not more, than mitosis, and I will tell you why. Well, the why is to create these genetically unique gametes and thus genetically unique organisms. So let's write to create genetically unique organisms. Now, why might genetic diversity, biodiversity, genetic uniqueness in a population of birds, population of whales, population of strawberry plants, you name it, why is it so important to be different genetically? What would you guys say? Natural selection, absolutely. So, oh, I forgot, at Meiosis we have discussed evolution already, so this is perfect. In a population that is diverse and we have varying genes among us or humans or whatever organisms, it is less likely that a pathogen, a disease-causing organism, or a predator can wipe us all out. One of the classic examples is the potato famine in Ireland. So during the potato famine, there was a fungal pathogen that just completely decimated all these potato plants. And it's because they were the same exact strain, same exact genetically susceptible species of plant. And it was able to just wipe them out. But as long as there's one resistant, different organism to that disease-causing organism, then that different plant or animal is able to survive. Hopefully one other survived, reproduce, pass on those resistance genes, and then the new population that arises is resistant to any future pathogens of that kind. So essentially having this heterogeneous, very diverse population of organisms is always favorable, because there's always a chance that you'll have a beneficial adaptation among that population that will allow for survival against any kind of predator or disease, etc. So it's extremely valuable to be unique. Okay. Does that make sense? Okay, so how? Well, the how is PMAT, again, 1 and PMAT 2 for meiosis 1 and meiosis 2. So PMAT 1 and PMAT 2. And that's what we're going to diagram out and discuss today and next time. All righty, any questions before we continue? Okay, cool. I shall wait. Phase 1, that is what we'll be talking about. So the why is to create new, unique organisms. Yeah, to create different, genetically different organisms in a population, yeah. As opposed to mitosis, where you're in asexual reproduction, we're just creating two identical clones, really, from mitosis. But we're going to look at it right now, actually, speaking of that, how we get this uniqueness through meiosis. And meiosis is why you don't resemble your parents exactly. It's why you don't resemble your siblings exactly. Even though you have similar DNA and you have commonalities, totally, we're going to look at why you're still different from them. And the first phase of meiosis is prophase 1 of meiosis 1. And I hope you guys have your colors ready, because specifically blue and red, we're going to be doing some coloring of sorts. Okay, So let's write prophase 1. And I just want you guys to keep in mind that this germline cell that's going to undergo meiosis also goes through interphase before meiosis, and it has a cell cycle. So I'll just write that. Interphase occurs prior to meiosis. Meiosis. And as we know from, that looks kind of small. As we know from learning about interphase, S phase of interphase, the chromosomes have duplicated. All of the DNA has replicated, right? And so we'll have sister chromatids when the condensing occurs during prophase. So I'll write DNA replication occurs, or takes place, right? Okay, so we're going to see that during meiosis, there are some similarities to mitosis, and there's also some differences. And in prophase 1, let's do the big cell. Hold on. Actually, that's fine. There we go. Again, we have 46 total chromosomes, right? 2 sets of 23, but I'm gonna do 4 total, just like we drew out with mitosis, because it would be too much to draw. So I'll do 2 homologous pairs. Let's do. There's 1 pair, and let's do another pair here. Okay. So just like in prophase of mitosis, the nuclear membrane is going to dissolve. It's going to break down. So nuclear membrane dissolves. And then also, like in mitosis, the chromosomes are formed. You could say condense and are visible. So that's exactly like we saw during mitosis. And then I'm going to point out some unique differences here that occur during prophase 1. One more thing, of course, we have the spindle fibers. The spindle complex is going to form on either end of the cell. So we'll have these form. Let's also label this as a germline cell. and it is diploid. It's very important that we remember this is a diploid cell, 2N, right? We use that to denote for a diploid cell. And in this example, the diploid number, 2 sets of 2 to make 4 total. So 2N equals 4. Okay. The first thing that occurs that's very unique to meiosis, and that scientists actually don't know why, not why, I'm sorry, they don't know how this happens, but it does, and it's extremely important, as we'll see later on. But during prophase 1 of meiosis, the homologous pairs find each other. Notice how I drew them really close to one another? So our homologous pairs sort of match up. Meaning the 2 chromosome 1s come together, the 2 chromosome 2s, the 2 chromosome 3s, the 2 chromosome 4s. They all just find each other and they come together. It's pretty amazing. It's like, how does chromosome 6 know not to come next to chromosome 1 and line up with him? It's so scientists still don't know how, but we know it's very important for the future or the rest of this process. And of course, in mitosis during prophase, they don't do this. The homologous pairs, they're all just kind of jumbled together. They're not like pairing up with one another. Okay. The next important difference that occurs is called crossing over. So, do an asterisk here. Crossing over. And this is when, and we're going to draw this, visually represent it, but this is when homologous chromosomes exchange large segments of DNA. Large segments of DNA. I'll sort of draw out what that looks like. Okay. So let's draw a crossing over event between these chromosomes here. Now, recall that these are sister chromatids for this chromosome, right? Identical strands of DNA. These are also sister chromatids for this chromosome, genetically identical. Crossing over occurs between non-sister chromatids of homologous chromosomes. So this is a homologous pair, right? 2 homologous chromosomes. When I say non-sister chromatids, I'm referring to these 2. separate chromosomes, they're actually going to break off a piece and swap with one another, like this. And so that's what we call crossing over. It's gene swapping between these non-sister chromatids of these homologous chromosomes, these different chromosomes, that their non-sister chromatids kind of break off and reattach and they switch. It's part of generating this diversity of genes. Crossing over, it only occurs for non-sister chromatids. Remember, sister chromatids of a chromosome are attached to one another, right? But see how these non-sister chromatids, they're not attached to one another? They swapped, right? So we have some blue here now and some red here. So those are the chromatids that will do the crossing over. And crossing over happens between, I think the statistic was like, on average, like 13 pairs per cell division out of the 23. So it happens quite frequently. Okay. Now, that's it for prophase 1. You saw I took a picture because I have to remember what these look like. So I'm going to erase inside here, but I would draw another cell for the next phase. Since you don't want to erase this information, I would just draw another cell for metaphase 1 as the next step in meiosis 1. Okay. By now our nucleus has dissolved away. We enter our next phase, metaphase 1. We are still diploid. I'll just keep that there. Keep that. Get rid of this. Okay. And now we're at metaphase 1. Still part of meiosis 1. Again, some similarities to mitosis, some differences. Okay. So I'm going to redraw the chromosomes now for metaphase 1. See my picture here, what they look like. Okay. And then I want you to take a look at how I draw them. Don't do that. Remember, there was a crossing over. There we go. And homologous, so they should look the same size. All right. So during metaphase 1, homologous chromosomes, align side by side. And I'll add in the center of the cell, right? So let me add in the center of the cell. So again, our chromosomes are, move my phone here. Our chromosomes are lining up in the center of our cell in metaphase, just like we saw before in mitosis, but I underlined side by side. Why? I will show you. I want you to recall that during mitosis, so during mitosis, kind of a flashback here, specifically metaphase of mitosis, chromosomes lined up how? How did they line up? They lined up in the center, but how? Linear, vertically, single file, one by one, there exactly. So it was like this, right? Notice how I drew those. The homologous pairs are lining up in the center, side by side. This is going to be incredibly important in meiosis for two reasons. The first being, meiosis is known as reduction division. Because they're going to line up side by side like this, at the end of meiosis 1, we're going to find that our two new cells have less chromosomes than they began with. They lost genetic material. And it has to do with them being side by side. So if you think about it, if I have 23 pairs here, 23 rows, 46 total chromosomes, and then I separate them, well, now we have 23 on this side, 23 on that side, right? And so our new cells will only have 23. But if we have 46 of them lined up like this, each new cell will have 46 at the end of the day. All that genetic information is retained in the two new cells after mitosis. But we're going to see that after meiosis 1, there's going to be a loss of genetic information in our two new cells. We will no longer be diploid. So just keep that in the back of your head as we go through this. Of course, to orient them in the center, our spindle fibers are doing this again. And then besides lining up side by side, one other very important thing here that occurs. And it's called independent assortment. So independent assortment occurs. We're going to really kind of dive into what that is. Let's see here. Does crossing over only happen in one? No. So, and remember, it's non-sister chromatids, right? Between two different chromosomes. It only happens during prophase 1, right? And then it can occur, really, I think it's random, but it can occur in like up to, it can be anywhere from 13 pairs to 20 pairs, 23 total pairs, right? And crossing over, I always forget the statistic, but definitely a lot of them will do crossing over. A lot of the pairs will. I only drew one for simplicity. Otherwise, we'd be here all day. But yeah, this can occur in a lot of the homologous pairs. Okay, so independent assortment, what does that mean? So like I mentioned, our homologous pairs are lining up side by side, but which side they line up on is completely random, completely. So I could have drawn this homologous chromosome here and this maternal homolog, to be more specific, over here. Totally random. So where they line up in metaphase 1, along this, what we call the metaphase plate, just the center here. So which side they line up on for all the 23 pairs is random and independent. So of course, there's so many combinations. There's so many ways they can line up. And we're going to explore why this is so crucial to producing genetically unique gametes. We're going to actually take a step back from this process and look at independent assortment up close. And to visually represent it here, I'll kind of just do this arrow. Like these could have swapped places, could have drawn it here or there. But the next thing I want to do is practice with independent assortment. So I'm going to erase this now. We're going to practice with independent assortment and give a more clear definition as well for your notes. Independent assortment. Okay. So independent assortment refers to the independent or random arrangement of homologous pairs at metaphase 1, metaphase 1, kind of ran out of room there. Okay. Do a comma, resulting in different possible genetic combinations. Genetic combinations in the gametes. independent arrangement of homologous pairs at metaphase 1, resulting in different possible genetic combinations in the gametes. So what do I mean by this? How is this happening? Let's take a look. Just for an example purpose to demonstrate this idea, let's look at 3 homologous pairs in a cell. So this is all during metaphase 1, right? So they're aligned side by side. And the orientation of which side they line up on is random. But for this example right now, I'm going to do it like this, where this chromosome is our chromosome 1 from mom. So I'm going to do 1 with an M, like that. This chromosome's 1 from dad, or paternal, whichever. This is our chromosome 2 from mom, chromosome 2 from dad, 3 from mom, 3 from dad. So I arranged them like that. Okay. Now, if I split this cell in half, which eventually is going to happen, split it right down the middle here. We're going to end up with 2 new cells, right? So 2 new cells. And as we see, if I split it in half, one side has 1M, 2M, and 3M. The other side has 1D, 2D, and 3D. So we have 1M, 2M, 3M, 1D, 2D, 3D. Okay. Now, I'm going to switch these two, okay? I'm just going to go ahead and switch the position of those. So visually represented, if I switch those, then I would have the paternal here now and the maternal on this side. Independent assortment means they align randomly. So that could have happened. Oh, well, now I have 2 more combinations, don't I? When I split them down the middle. So now I have 1D, 2D, 3M. Do that here. On this side. And then on this side, I have 1M, 2M, 3D. And etc. I could have switched those, I could have switched those, and essentially this is here are the range of possibilities. And then just draw them out for you. Running out of room. Let me redraw that guy. not much better, but whatever. Okay. So I did 1M, 2D, 3D, 1D, 2M, 3M, 1. Okay, I think that's all the combinations. Move that up. Okay. So based on where these align, I can end up with 8 different genetic combinations once this splits, this cell. This is with 3 homologous pairs. So we can even write 3 homologous pairs and that can give you 8 combinations. But in reality, there's 23 pairs. So we're going to do a cool little example with that. 23 pairs that are swapping places randomly. Okay. So let me go to my slide first. Oh. Well, remaining phases. First, I want to do something before we get to the remaining phases. Show you an interesting thing with independent assortment. It's 23 pairs. Okay. Now, when we had 3 pairs, we had 8 possibilities, right? Genetic combinations. When you have 23 pairs, the equation is 2 to the 23rd power. So 2 times 2 times 2 times 2 times 2, 23 times. So that's how many genetic combinations are possible in, say, producing the egg cell in a female. An egg cell. So 2 to the 23rd is roughly 8 million combinations for producing that egg. Now, the egg combines with the sperm cell to form the first cell of your new organism. If you're making a new human, a new plant, a new mushroom, fungi, right? Sexually reproducing. We're combining these different sets of genetic information. And so you have 8 million options for the egg, multiplied by 8 million options for the sperm, all because of this independent assortment. So 2 to the 23rd genetic combinations in sperm cell, again, is roughly 8 million. I'll abbreviate it, ran out of room. So this is 64 trillion possibilities. So let's write 64 trillion genetic possibilities. And really what this means is to create you is a 1 in 64 trillion chance. There were a lot of other options. So we're all very unique, is what I'm saying. So it's pretty amazing. And it's even more interesting that, I know, Woe was right. This doesn't even include crossing over occurring. I mentioned crossing over can occur on 13 or more pairs in each one of these individuals as this process occurs. And if even one crossing over happens, that's another possibility. Because it couldn't have happened, or it didn't have to. It could have not happened. So it's another variation. So it's just an incredible, unique, system. It's a system of producing genetic difference among organisms that's been retained for a very long time. Okay. Questions about this before we move on to anaphase 1? All right, cool. Okay, so anaphase 1. So that's metaphase 1. Independent assortments occurring during metaphase 1. They're lined up in the center, side by side, these homologous pairs. Now we enter anaphase 1, still meiosis 1, right? And really the story for the rest of meiosis 1 is quite similar to mitosis, nothing too unusual. But of course, this time our chromosomes are side by side, and they're going to be pulled to opposite ends by with the spindle fibers. So let's visually represent that, draw it out, write the definition. See what my chromosomes look like. So remember, we're still 2n = 4, still diploid. Just picking up where we left off. And we've got Not the best artist, but should do. Chromosomes are being pulled to either end of the cell by our spindle fibers. This. And so during anaphase 1, homologous chromosomes are pulled to opposite ends of the cell. All right. Now that our chromosomes are pulled to opposite ends, we enter telophase 1. Again, draw a new cell. I'm just gonna erase here. Actually, I'm gonna draw a new cell myself because, as we know, during telophase, cell gets a little longer, starts to pinch in a little bit, so it's gonna look like this. We're gonna have 2 nuclei start to form at either end. Draw my chromosomes here. Like that. So telophase 1, 2 nuclei form at either end of the cell. Really the main event here, end of the cell. Like this. And last thing that occurs during meiosis 1 or following meiosis 1 is cytokinesis 1, where cytoplasm divides and splits into 2. And we get 2 new cells. Before we enter meiosis 2, which will be the subject of next class. And then, of course, our nuclei has fully formed at this point. This is cytokinesis 1. And as a sort of final thought here, if I had 4 chromosomes to begin with, and now each of my new cells only has 2 chromosomes, what would I call these cells? Their chromosome number was reduced by half. So each one only has 1 set, as opposed to 2 sets. They are haploid. That's exactly right. So they're no longer 2n equals 4. Each one of these cells is n equals 2. So by the end of meiosis 1, we go from diploid to haploid. And for some reason, I like to do this. I write haploid in all caps with a bunch of exclamation marks. I saw a professor do that once. I was like, yeah, I'll remember that. So by the end of meiosis 1, we have reduced our chromosome number from 2 sets to 1. So really, at the very beginning of a human cell undergoing meiosis, we have 2 sets of 23. And after meiosis 1, each new cell has 23 only. So they have lost information. They have lost chromosomes. They only have 1 set each. They are haploid. So this is where we'll pick up next time. As we talk about meiosis 2 on Thursday. So any questions before we take a break and then talk about, well, not the lab, but a genetic disease project that we're gonna, that you're gonna do. That's due later. We'll talk about the details. 23 chromosomes per cell. Yes, correct. In a human, yeah. Here in my example, it's 2 because we started with 4. But yeah, if I were to really actually draw this out, which is pretty tough on a small whiteboard, each one would have 23 X's in it. Opposed to I think I know what you mean, but opposed to like mitosis. Oh, right, exactly. So in mitosis at the end, there's still 46 chromosomes in each cell. They're single-stranded, so don't get too tripped up on this because it can get kind of confusing. But essentially, because during mitosis, the chromosomes line up like that, you're going to have 46 of them that separate. So each new cell is going to have 46 units of information, meaning each new cell has 46 chromosomes. Here, because they lined up side by side, like that, you only have 23 rows, 46 total. And so each new cell is going to have 23. There's going to be information lost, these chromosomes, because of how they lined up in meiosis during metaphase 1. So each new cell here would end up with 23 chromosomes. When they line up single file, each new cell is going to have 46 single-stranded chromosomes, but still 46 unique sets of information there. Yeah, it does depend on how they line up. Absolutely. Yeah, I try to emphasize that point by taking really a whole lecture to look at metaphase 1 and independent assortment. Awesome. All right, so let's take a 10-minute break. Today's lab mini lecture won't be too long. I'm just going to kind of briefly go over this genetic disorder project. Yeah. So what time is it? 4:20. So 4:30. Meet here at 4:30. Oh, journals. I finished grading them, so I have to input them on Canvas. So I've been super busy this week making an exam for my other school because they have an exam tomorrow. So once I finish that, I should be able to put them on Canvas for you guys. Yeah, I took the whole weekend though to finish that. Yeah, so they'll be up soon, I promise. And then you can hate me. No, I'm kidding. I loved them. They were great. So, okay, so let's come back here at 4:30, 4:31-ish. We'll chat. And let's play a fun song. Oh, yeah. Great song.